And today we're going to finish our discussion of the neuroendocrine system, and we're going to focus on three key absolute functions of the neuroendocrine system that you can add to all of the pituitary functions we talked about in the last week. And as you can tell, because there are so many hormones, so many functions, I mean, we're dealing with a bunch of different organ systems. I really don't like in-class exam questions over this. So I'm happy to help you guys work through some of those questions and know that I'm going to be giving you take-home exam questions, which I've told you before. Okay, so let's start with the regulation of temperature. And basically the integration of the hypothalamus is vital for temperature control. And we, as you know, we receive information from the periphery, which causes our body to change, either sweat or shiver in response to cold, for example, or sweating in response to heat. And that sensory information gets imparted to a set of neurons up in the hypothalamus, and they're up in the paraventricular nucleus. So you remember where that is, right outside the third ventricle on either side. And these neurons produce a hormone called thyrotropin releasing hormone, or TRH. And TRH is produced by the neurons and then sent in packages in little granules down to be released in the stalk median eminence capillaries here. And there it's collected by the portal veins, which go down to the second set of capillaries in the anterior pituitary. And there are special receptors called thyrotropes or TSH cells. Thyrotropes receive the TRH, and then they will secrete thyroid stimulating hormone, or TSH. Now, thyroid stimulating hormone is easy to figure out by its name. It stimulates the thyroid gland. And one of the things we need to do when we get cold is to increase our metabolic rate. And this is what the thyroid gland does. It produces two hormones, thyroid hormone T3 and T4, that will increase basal metabolism and get our metabolic rate functioning so we can respond to cold. And of course, these hormones are vital to feed back, and they will feed back to the pituitary thyrotrope, shutting it down when there's sufficient stimulation. And they'll also feed back to the TRH neurons, shutting them down when there's sufficient stimulation. So that's the circuitry. And just reminding you, if I ask you to describe the circuitry, this is basically what you do in words in an exam question. You would tell me where the TRH neurons are, what they produce, where they secrete it, and then how it's taken to the pituitary, what cell types receive it, and then what those cell types secrete, and then what they do in the periphery. In this case, you have stimulation of the thyroid gland. So I've had students skip elements here in the circuitry when I've asked a question about, well, how do we respond to cold? So just make sure you've got all of those elements in the circuitry. Okay, this is just another picture of the periventricular nucleus. And the new information here is that the subsets of neurons in the periventricular nucleus that does this are small. So they call them parvocellular, small neurons. And basically, again, this just shows the same picture. You see the axons going to the stalk median eminence and secreting into the anterior pituitary through these portal vessels. And these are actual pictures of TRH neurons and there's the third ventricle right here. And basically, these are the TRH neurons. This happens to be a neat picture in an embryo. So E18.5 PVN, so this must be a mouse. And basically, you can see that they develop very early and as needed. All right, again, this is a great picture to show the blood supply, and this is the last time you'll see it, but it gives you another review. So here your TRH neurons are at the top, and this is the capillary plexus in the stalk median eminence region where they secrete. And then this is a nice picture of the portal veins, which are carrying, basically connecting this set of capillaries to this set of capillaries. And this set has the TRH in it, and then it carries it to the anterior pituitary. So hopefully repetition helps you understand the entire relationship between the circulatory system and the nervous system and the endocrine system. So this just shows you the feedback loop, negative feedback. I've already told you about it. Thyroid gland produces thyroid hormone. And when the thyroid hormone levels are high enough, negative feedback shuts down TRH and TSH. And then when thyroid hormone levels drop, the TSH cells can be stimulated again. So the bottom line is it's a complete intact system here, the whole circuitry, and complete with negative feedback. And this is a picture of the thyroid gland, and you can see it's got just a single layer of cells here, and these cells will grow taller when they produce thyroid hormone. And they're a very interesting set of cells, and we don't have time to really study them in detail, but the bottom line is that they produce their thyroid hormone and they store it in a huge molecule, a huge glycoprotein molecule called thyroglobulin, and they store it out in the colloid. So thyroid hormone is a really small, it's just 2 amino acids, 2 tyrosines that are iodinated and hooked together. That's all it is. And yet they go to the trouble of putting multiple sets of those hormones in this big glycoprotein, and they store it out here in the colloid. And then when we need thyroid hormone, they'll pick this up by receptor-mediated endocytosis. They'll pick up the thyroglobulin, break it down, and then they secrete it into the bloodstream. So that's basically the mechanism. And of course, all of that is stimulated by TSH, thyroid stimulating hormone. So here's a normal thyroid a follicle, and it just shows you the steps that it takes to make thyroglobulin, and it'll iodinate it so that your T3 and T4 are out there stored in the form of this protein. And then in a stimulated, let's jump over here, you'll have a TSH stimulation and you'll have thyroglobulin breakdown. So the cell will pull it in and break it down in lysosomes and secrete the T3 and T4. Now, one of the things that can happen is if you have a problem with your thyroid, it will shrink actually. And the other thing that can happen is, let's say you can't make T3 and T4. Let's say you can't put iodine on it, for example. Then you're going to have no negative feedback. And this story is a story of how you make a goiter, because the TSH doesn't know, pituitary doesn't know what the problem is. All it knows is that there's no negative feedback. So this will cause the thyroid cells to grow and grow and grow and eventually cause a goiter. So that's how a goiter is formed. Now, all the hormones I've been telling you about have rhythms, and diurnal or circadian. In the case of TSH and TRH, they work together. It is diurnal. That means that light affects it. And so normally you'll get pulses of 90 to 180 minute intervals. You'll have a big pulse. However, as you approach nighttime, and here's nighttime here, right here, here's midnight. You're going to get more and more and more pulses out. And it's really cool because, you know, you're not moving around and your body could get really cold at night. And in fact, that's what this is for. It's to keep your body temperature up. So here, this shows that men are a little hotter than women here. And, but this is just due to higher level of pulses of TRH and TSH, which increases your body temperature. And it's probably why you throw your covers off in the middle of the night, because your body is actually getting hotter, and as it should, to keep you from freezing to death, I guess. So, so the bottom line is that this is diurnal. It is driven by light. So the suprachiasmatic nucleus signals to TRH neurons that, okay, the sun has risen, so get started with your day. And then when the sun goes down, there is a signal that causes this. And I'm not sure exactly what the signal is, but it is a light dark signal. The absence of light causes the TRH and TSH to go up. Now, thyroid hormone is really important for a variety of functions. It's absolutely vital for growth. And if you don't have thyroid hormone, also your pituitary somatotropes won't function. And so they'll actually go away. They'll involute. It's really, really weird. It's important in brain function and brain development. And it's vital in frogs, for example, for metamorphosis. So it's very, very important for life. And if you have pituitary deficiency, one of the things you absolutely have to do is replace your thyroid hormone. Okay, so one of my favorite questions: trace the events that occur if you step into a cold environment. And supposing you, I'm going to take that phone and get rid of it. All right. Okay, so usually my husband takes care of that, but he's not here today. So one of my favorite questions is to say, let's say you fall in an icy lake or something like that. What is your brain going to be doing? Well, it's easy. You say to yourself, well, the first thing, I'm going to feel the cold, and your sensory cells will react and send messages to your brain that you're cold. And of course, your system will start to respond. You'll start shivering and you'll do everything you normally do. But in order to get your metabolic rate up, you've got to activate your TRH neurons. So basically, immediately think paraventricular nucleus, small neurons, parvocellular neurons, and that produce TRH, thyroid releasing hormone, or TSH releasing hormone. And basically, the TRH then will be secreted into the pituitary bloodstream. So be sure you talk about where. And then it goes to the anterior pituitary and stimulates the thyrotropes. So this is a good question to practice with, and just go back through the circuitry. Now, the rhythm of secretion, well, it's highest at night, and it is diurnal, so it is dependent on light and dark. And then the functions of thyroid hormone, mainly to increase metabolism, but it's also important in growth and development and metamorphosis. Okay, now let's look at a completely different part of the pituitary gland, and that is the pars nervosa, or the posterior pituitary. And this is really an interesting subsection of the pituitary, because it really developed as a downgrowth from the diencephalon, or the hypothalamus region. And it's just a bunch of axons that grew down. And along with them came some supportive glial cells. And they're just sitting out here in a process, a big old neuronal process called the posterior pituitary. And that's all that is. And so if you trace them all the way back, you're going to find their neurons, their cell bodies up here, either in the paraventricular or the supraoptic nucleus. Either one of these nuclei will contain those neurons that send their axons all the way down. The other thing about it is that it has a separate blood supply. It's got its own blood supply. It's not dependent on the blood supply to the anterior pituitary. So it just develops from the inferior hypophyseal artery, which breaks up into capillaries right here. So why would it do that? Well, you can figure that the hormones are probably important enough to get out of the blood-brain barrier. And so this is outside the blood-brain barrier. The glial cells in this region are supportive, but they're not going to prohibit things from reaching and stimulating these vital hormones. So the other thing about these neurons is that they're large. Remember, I told you the TRH neurons are parvicellular. Well, these are magnocellular. These are the largest neurons up in the paraventricular and the supraoptic nuclei. Okay, so what do they produce? They're separate neurons that produce each of these two. One set produces vasopressin. You can also call it antidiuretic hormone, ADH, and it's very important in water and salt balance, osmoregulation. So it would maintain our water balance, our salt balance. I'll give you a good example of what might stimulate it would be if we were to have too much bleeding, hemorrhage, something like that. That would immediately trigger vasopressin release to shut down blood flow and to preserve water and fluids and keep us from basically dying as long as it can. The other hormone that's produced in a separate set of neurons is oxytocin. And oxytocin is really interesting. It has a number of functions related to social behaviors we'll talk about in a minute. But initially, it's most famous for childbirth. And remember, I told you that oxytocin is released when the baby's large enough and starts pressing on the uterine wall and the cervix, and that will exert positive feedback on oxytocin. Oxytocin is also released at the time of suckling, lactation. And so what oxytocin does is squeeze the ducts of the mammary gland so that milk can go into the ducts, squeezing cells around the gland itself, just to squeeze the gland to put milk into the ducts. And if you don't have oxytocin, you could produce milk, but you cannot lactate. So you absolutely need it to eject the milk. And mothers who have had a baby will call this milk letdown. You can actually feel it. And so basically all of that is due to oxytocin. So again, this is just another picture of paraventricular nucleus and the supraoptic nucleus. And of course, you know where this is. All you have to do is look at the name. It's above the optic tract. So there's the supraoptic nucleus. And these are the axons, which are heading down, and they form the posterior lobe. And that's all the posterior lobe is. It's just a bunch of axons and some supportive glial cells. And then, of course, you have the capillaries they secrete into. And so, as I said, oxytocin is important for uterine contractions. And the mammary gland, they're called myoepithelial cells to squeeze the milk into the ducts. And then vasopressin is very important. It raises blood pressure and it conserves water and sodium. Now, you can take your time to read this. This actually describes in more detail what I just said about oxytocin release. And basically, the feedback is mostly positive. And so I've already talked to you about pressure on the uterine wall, feeding back positively to neurons, which then feed back to the oxytocin neurons. And then they release their oxytocin. And so that stimulates labor. In the case of suckling, all the baby has to do is grab onto the nipple and start to suckle. And that sends a neuronal signal back up all the way to the brain and to the oxytocin neuron, and says, okay, we need to get milk into the duct so the baby can eat. And basically that's the so-called milk letdown. And if the baby suckles on one breast, there'll be milk letdown in the other breast, as nursing mothers know. So basically, this again is positive feedback from a neural stimulus, and here positive feedback from pressure on the uterine wall. Now, the vasopressin is dependent on structures called baroreceptors, and these are going to test blood volume, blood pressure, and they're in the wall of vessels and also in the heart. And when there's a drop, like for example in hemorrhage, vasopressin will be stimulated to be released. And so basically it's a direct neural circuitry there. Also, vasopressin senses, and there are sensors for plasma osmolarity. And so if we need more water, vasopressin will in fact, or if we need to conserve water, if plasma osmolarity goes down, vasopressin then is stimulated. And it is, its targets are in the kidney and also arterial smooth muscle. So for example, it'll constrict the arteries to allow the for a more I guess control of blood pressure at this point. And then it also regulates water conservation and sodium conservation here in the kidney on specialized cells. And again, we don't there's an awful lot of physiology involved, and we don't have time to talk about the details, but just know in general what its regulation and what its function is. These are pictures showing immunolabeling for oxytocin. I think this is probably up in the paraventricular nucleus, and this is vasopressin. And basically, for the most part, as I said before, they're in separate neurons. And every once in a while, you'll get some that have both, but these are basically, this is just the immunolabeling for the oxytocin or the vasopressin. And these are higher magnifications just to show you that the labeling is kind of granular, because the oxytocin and vasopressin are stored in secretion granules, and then they're sent down the axon to be released in the posterior pituitary. And when you get to the posterior pituitary, you can see secretion granules, and they're sitting in nerve endings, basically, that are just sitting on a capillary. So here's a capillary. This is an endothelial cell. And so they will be released by exocytosis into the capillaries. And basically, that's basically what the posterior lobe looks like. It's just got a lot of nerve endings with these secretion granules in them. So basically, quizzing yourself. So identify the sites of production, the route to the final storage site, and the site of the storage site. So know where oxytocin and vasopressin are produced. They're not produced in the posterior lobe, they are only stored there. So this is, again, a good question that I like to give, and just be able to go through and identify these sites and where they're secreted. And then basically, this is another way of asking the question: if the fiber tract in the stalk is cut, what hormones would you expect to be reduced? Well, you're going to say oxytocin and vasopressin, and that can happen. Um, and then define the function of oxytocin and its positive feedback regulation. So don't forget that it works on both these myoepithelial cells in the mammary gland as well as the uterus. And and then the function of vasopressin or antidiuretic hormone. It's regulated by baroreceptors and information about plasma osmolarity and blood pressure. And so when blood pressure drops, this stimulates vasopressin to squeeze the arterioles down, to increase blood pressure and increase conservation of water and sodium. Okay, so I hope that's been clear. And let's now move to the final section, which is appetite regulation. And so when we think about appetite regulation, there are a lot of factors involved, and it's gotten a lot of excitement over the last, 20 some years because of the discovery of leptin. And so the endocrinologists just grabbed on to appetite regulation once leptin was discovered. And so it's a huge area of research. But there are a number of things that motivate us to eat and help us eat. And of course, one of the most important is depleted energy stores. So if you basically are exercising a lot, that's going to signal that you need to eat. If a satiety signal that makes you feel full goes away, you're going to want to eat. And then, of course, you've got this learning habit, social pressure. If you are at a cocktail party and people send wonderful trays of food your way, you may not be hungry at all, but you may find yourself eating no matter what. And then caloric homeostasis. This relates to individual cells and their state. And all of that is added up to really signal depleted energy stores as needed. So all of these factors end up signaling to the neuroendocrine system. And I mean, you can sort of figure here, there's a lot of emotional components too, social pressure and learning and things like that. So they have known for a long time that the hypothalamus is involved in appetite. And one of the coolest early experiments was that they'd go in there into rats or cats or whatever, and they'd lesion the ventral medial nucleus. Remember, here's a medial nucleus and it's ventral, so that you know where it is. Here's the third ventricle. And they discovered that if they lesioned that, it causes obesity. So the animal got really fat. So they thought, aha, maybe this regulates. And then they would go along and they would lesion the lateral hypothalamus. And that caused starvation. The animals would just stop eating. So they go, well, the whole appetite regulation system must be in that. Well, that's definitely an oversimplification, but it does describe some of the early studies. So what happens when you do both? Well, you've got your obesity going on here with the ventral medial, but then if you go ahead and you do the lateral hypothalamus also, you reverse the obesity and you cause starvation. So it's kind of confusing, but these are in the early days. These are some of the early things they used to do to play around. And here's another fairly early experiment. They attached rats to each other. They joined their circulatory systems, and they stuck a lesion in the ventral medial. And remember, the ventral medial causes obesity, okay? And so, of course, the rat with that lesion became hyperphagic. That means it's eating too much and obese. But the rat without the lesion became lean, suggested something secreted by the lesioned rat affected his or her eating habits. Okay, and so something's going on here, and it's in the circulatory system. So for many years, people have been looking for that something, and it wasn't until the mid-90s that this was actually defined. And they defined it as leptin. Now, leptin, the concept of leptin was known, as I just said, basically in 1950, Jackson Laboratories discovered this OB mouse. And basically, they knew there was something wrong, but they never really were able to define the gene until Friedman and his colleagues in 1994. And basically, they were able to discover this factor. And they discovered it was produced by adipocytes or adipose cells. And if you ever studied histology and taken a look at adipose cells, you'll say, wow, you know, it seems like a simple cell. Why look for endocrine factors for it? And I think people just underplayed adipose cells for years and years and years. But it's a very important set of cells in our body. And one of the things it produces is leptin. And so we are assuming that leptin target cells in the ventromedial nucleus of the experiment I just told you were destroyed. And so, but that leptin was high. And especially as the animal ate and became obese, because leptin is high in an obese animal. And so then the animal connected by parabiosis basically ate less, because the circulating leptin rose in the obese animal and got sent over to the connected animal, and became very, very lean. So Friedman figured that was the factor we were looking for. And so here's the OB mouse. Now, sometime later, they found mice that lacked the leptin receptor. And these are now called DB mice. So we have OB mice and DB mice. And the other thing to know is leptin, of course, as I said, are produced by adipocytes. But what you may not know is Angela Odell knocked out leptin in several cell types. She knocked it out in adipocytes, and she also knocked it out in pituitary somatotropes. But the cool thing is that she discovered that when she knocked it out in adipocyte, there was virtually no circulating leptin, absolutely none. We were thinking maybe some of it would come from leptin from other cell types, but it doesn't. The major circulating form of leptin comes from adipocytes, and that discovery was made by Angela. Okay, so people played around with these OB and DB mice, and so you can kind of take a look at this. Again, we're parabiose, and so you connect a lean mouse to an OB mouse, and of course, there's no leptin here, and there is leptin here. And so connecting the circulatory system will cause this mouse to cure this mouse. So that's one thing that happens. Now, the DB is out of luck for two reasons. Both mice are out of luck. First, while this mouse has plenty of leptin, the DB mouse doesn't have a leptin receptor, so cannot respond. And sadly, though, the DB mouse has lots and lots of leptin, which goes to this mouse and causes this mouse to undergo starvation. So this is not a very pleasant parabiosis experiment. And then, of course, if you connect an OB and a DB, you can pretty much guess what's happening. Okay, there's no leptin here, but there's lots of leptin here. So this mouse is going to cure this mouse, but this poor mouse is out of luck because it cannot use its leptin. So these are some of the factors that people have played with, and of course, leptin explains all of these changes. So now, how does it work? Okay, and so before we go into details about the sites, we need to define eating patterns, eating habits, whatever. And so you'll be able to say, okay, I know what orexigenic means. Orexigenic means increased eating. And orexigenic is also muscle building. So if you're in an orexigenic state, you're eating more, you're building muscle, you're slowing your energy expenditure, because the idea is you want to build your resources, and you're going to increase obesity. So everything that promotes that increased eating may lead to obesity. Now, anorexigenic is just the opposite. You have decreased eating. So leptin is anorexigenic, for example. And also it's a catabolic state. You're breaking down fat. And I'm not saying you're breaking down muscle, but you're definitely breaking down fat. You're increasing your energy expenditure, increased metabolic rate. This is one of the things leptin does, is increases metabolism and decreased obesity. So the bottom line is we have to keep in mind that there are two sets of hormones. One set is orexigenic and the other set is anorexigenic. So what are some of them? Okay, well, let's name them. Among the orexigenic peptides that drive us to eat are neuropeptide Y. It's a very famous one. And it's produced up in the arcuate nucleus of the hypothalamus. Okay, so that's a nucleus that's just above the pituitary. And it's high just before mealtime. It remains high as long as we don't eat. And its target neurons are, it sends signals to cells in the paraventricular nucleus. And so if you have a lot of NPY, you end up with a state of hyperphagia, meaning you're eating all the time, and you can turn into a blimp. You can become obese. Another orexigenic is agouti-related protein. It's got a weird name, but it is co-secreted from the arcuate nucleus. And what it does is blocks anorexigenic peptides. And I'll tell you what those are in a minute. And so if you give AGRP, that'll stimulate food intake for up to 7 days. And again, both of these peptides can lead to obesity. Now, what about the anorexigenic peptides? Okay, they're also found in the arcuate nucleus. And the neurons, again, send projections to the paraventricular nucleus. And it's the paraventricular nucleus that regulates appetite ultimately. But these orexigenic and the anorexigenic peptides are produced in the neurons in the arcuate nucleus, and they receive signals from the periphery. And I'm going to show you the diagram in a minute. But just know that it's a two to three step process. So one of them are the very famous melanocortins, and the melanocortin alpha MSH basically, and it's got some really important receptors. There are MC3 and MC4 receptors, and they're critical in body weight regulation. In fact, they believe that this is going to be a site where we can actually regulate our body weight if they can get drugs that will affect these receptors. And as I just said, agonists of this receptor could help obesity. So alpha MSH. Now, one of the things that the agouti, AGR alpha MSH. So that's how it works. So another one is cocaine and amphetamine regulated transcript, or CART. It's again a weird name, but it is anorexigenic, and it is secreted from the same group of neurons that produce alpha MSH. We have no idea what its receptor is, but it is anorexigenic. Now, what you need to know is leptin stimulates. That's one of its major actions, is stimulates these anorexigenic peptides. And it inhibits the orexigenic neuropeptides. So it does double duty. So when leptin rises, let's say we've had a good meal, and leptin will be really high and will rise, and it'll inhibit us from eating more as it stimulates these guys, which then basically stop us from eating. So they're sent to the paraventricular nucleus, and the paraventricular nucleus then sends neuronal processes, sends axons out to basically all the neurons in our brain that will keep us from eating more. So here is a very simple model. You can see basically the anorexigenic peptides inhibit food intake, which is like a black box. And we're not going to go into details on the black box. And these neurogenic peptides stimulate food intake, and they all come from this tiny nucleus here. It's right on either side of the base of the third ventricle. You can see that here, called the arcuate. Now, there are other, I told you that the ventral medial is important in obesity. Well, some of these pathways that I'm talking about are in these other areas that inhibit appetite. And this is another one. This shows you more directly to the paraventricular nucleus. So let's, I know this looks complicated, but let's start here. Let's look at orexigenic. Let's just focus on the orexigenic pathway. Okay, so ghrelin. Now, ghrelin comes from the stomach. So when we have an empty stomach, ghrelin is stimulated, and it'll come out, and it will go to this and stimulate the orexigenic neurons to produce neuropeptide Y, AGRP, and AGRP will block the melanocortin receptor. You can see it up here. And neuropeptide Y will stimulate the neurons in the paraventricular nucleus. And I named some of them out here that basically stimulate us to eat. So that's the orexigenic pathway. Now, let's look at the anorexigenic pathway. Okay, so you've got some signals going on. Your glucose is raising, your 3 fatty acids, nutrients. Signals are telling leptin, okay, you're done with your meal. You really need to stop eating. And so leptin will come, and it will stimulate the anorexigenic peptides to produce CART and POMC. And notice I don't have any arrows from CART because we really don't know where it goes. At the same time, it's stimulating the anorexigenic. Look over here, it is inhibiting the orexigenic. So it does double duty, makes darn sure these guys stop stimulating or even stop getting produced. So it will shut down our eating. And so let's follow the anorexigenic peptides up. You've got POMC, and I told you about POMC when I talked about ACTH. In this case, the POMC molecule is in a neuron which has a different set of enzymes. And that set of enzymes will appropriately cleave to produce alpha MSH. And that alpha MSH will go and do 2 things. First, it binds to receptors in the paraventricular nucleus. Again, it stops us from eating. And then it also will go to the ventricle from eating. And you remember that nucleus is where if they did a lesion, then the animal got obese, okay? And so the bottom line is that's a very important set of neurons that receive this input to keep us from overeating. Now, there are a lot of other sites, and I don't have time to go into details, but there are the lateral hypothalamic nucleus, and I apologize, I'll have to look it up and then I'll email it to you. But the bottom line is the lateral hypothalamus is also an appetite regulator. And it may work also through the autonomic nervous system. So, but the main thing is the leptin-mediated circuitry here to the paraventricular nuclei. And I made a mistake if I inferred that these were orexigenic. They're not. They're anorexigenic. That's really important. That's an anorexigenic action here. So that's leading. I should color code that. That's leading from the paraventricular neurons. And so you look at oxytocin and you say, well, she just told us oxytocin stimulates labor and everything. Well, oxytocin is also a bonding hormone. It's important in creating social relationships and even relationships with your pet. And oxytocin is kind of a it's got a variety of functions, but another one is anorexigenic, which shuts down appetite. So I guess if you're building relationships, this might explain why you're not hungry. I don't know. If you ever watch The Bachelor or The Bachelorette, they never seem to eat when they're sitting at the table. They're always just talking, you know. At any rate, that's probably because of oxytocin. It's probably shutting down their appetite, but that's just my guess. Okay, I hope this is fairly clear. So as I said before, we also, because leptin is a metabolic increase in energy expenditure. So in addition to inhibiting appetite, you'll find it all over. It facilitates insulin action, helps us to have the appropriate glucose uptake. And one of the most important signals for leptin is starvation or fasting, because leptin is really dependent on, normal eating and everything, and a normal pattern of eating. So if you go into a fast, that will automatically lower leptin levels. And this will allow the orexigenic peptides to function and drive us to eat, and which is appropriate if we're starving. The big problem with humans, and this is something that is not true of mice, leptin will cause an obese mouse to get skinny. But unfortunately, obese individuals cannot go to the clinic and say, well, just inject me with a lot of leptin, doc, because the clinic will say, well, your leptin is already sky high. And the most important concept here is that in humans, obesity, well, I guess in animals too, the amount of fat you have equals high leptin. And your high leptin is just not shutting down your appetite. All of a sudden, you've become leptin resistant. And so they don't know at what point in obesity that an individual develops leptin resistance. And it is a real problem in endocrine circles, because they'd like to cure obesity. And unfortunately, the very hormones that are needed for the cure are no longer effective in these individuals. So it's a real problem, and the reason why they're looking for agonists of alpha MSH, for example, alpha MSH receptors. Now, I just want to spend a few minutes on the GI tract in the end to show you how our GI tract talks to our brain and also regulates appetite. And the first thing is insulin. And insulin has, when we're getting set up to eat, insulin has various phases. And the first thing that happens is that you have an increase. It's called you're getting ready to eat, and then you smell, and your insulin will rise. So your insulin is saying, oh, okay, I know what's coming, and I'm going to be prepared for an increase in blood sugar. And so then the food enters the stomach, and you have an even stronger rise here. So again, it's ready. It's definitely going to be ready for the breakdown, the food products which are entering the bloodstream, which would be glucose. So you would have a rise in glucose levels, and that, of course, stimulates insulin production. So this just shows you how insulin responds. And then the more clever is produce specific hormones that that signal the rest of the digestive tract to in response to what is being eaten. And these are all produced by these cells called enteroendocrine cells. So they're endocrine cells that are in the GI tract, and they're secreting. You can see a picture of one here. And they secrete into blood vessels. And they have a little process that extends up. They're almost like sensory, where they sense what is entering the system. It's kind of like, we think about taste bud receptors or something. So they're sensing what you're eating, what's going into the stomach. And they will secrete a specific hormone in response to the nutrient. So it's really kind of cool. And then they'll signal the brain and the neuroendocrine system, okay, she just ate a cheese pizza. We need to deal with a lot of fat, that kind of thing. So the signal, so this again is neuroendocrine. Remember, I told you neuroendocrine is defined by signals back up to the brain, just as it's defined by signals from the brain. So this just gives you an example. If you were to eat a cheese pizza, then there's a special kind of enteroendocrine cell that responds. It senses the fatty acids, and it stimulates it to secrete cholecystokinin. Cholecystokinin, or CCK, will then go to the liver to secrete bile to facilitate the digestion of fats. So it's a very clever way of signaling, oh, okay, you've got some work to do. Now, if you have a baked potato, you have a reporter of carbohydrates. And that reporter is found in the L enteroendocrine cell. And they'll secrete glucagon-like peptide, or GLP-1, and peptide YY. Both of those are important in insulin secretion, because of course the carbohydrates are broken down to glucose. And basically, you're going to need insulin so that we can don't have a glucose overload. These are also satiety signals. And so they may signal afferent nerves, which can go all the way up to those neurons that keep us from eating. So, so that's why we feel really full when we've had a baked potato. Another type of enteroendocrine cell will produce a pure orexigenic hormone, and that's ghrelin. And I talked about that earlier. That's a really important hormone that goes all the way up to the arcuate nucleus and stimulates AGRP and neuropeptide Y. And then there's another one called peptide YY, which is like neuropeptide Y, and it comes from the small and large intestine. So when you need to eat, basically, it will signal pre-meal hunger. And if you're fasting, ghrelin levels rise. Interestingly enough, ghrelin also stimulates growth hormone, but that doesn't mean we have growth hormone release every time we're hungry. But it is an important releaser of growth hormone. So those are two orexigenic hormones. Now the endocrine cells also will produce anorexigenic. And one of them is, I just said this before, glucagon-like peptide 1 with PYY will enhance insulin release and also inhibit food intake. Apparently it's not really strong, but it does do it. And the interesting thing about CCK, I already told you that it stimulates the liver to produce bile, but it also will signal satiety by sensing the stretch on your stomach. And so stretch receptors on your stomach will say, man, I'm full. And CCK is one of the signaling devices for that. And I guess you might, if you wanted to take CCK and leptin together, according to this one study, you could cause even more weight loss because they would work together. At any rate, so basically, these are the endocrine cells that signal the need to stop eating. And as I mentioned, gastric volume stretch receptors, these are all important. They signal via the vagus nerve and the area postrema. And as I've already talked about, cholecystokinin. And again, the vagus nerve gets involved, all reporting satiety. And then the other thing is caloric density. And it's supposed to play a satiety role. However, I just remind us that frequently, you know, a piece of pecan pie or chocolate cake even still looks good, even if you've had a big turkey dinner. So, but it's supposed to, you're supposed to take a look at it and say, oh man, you know, I am really full. I don't need that. But unfortunately, if your favorite aunt made it, you're going to say, oh, so you've got some social pressure going on. You're going to say, oh, yeah, I'll have a piece. So these are all the pathways I talked about. And I just want to impress upon you the fact that it isn't just leptin, it's coming from a variety of sources. And this just kind of summarizes. So you've got CCK, you've got mechanical stretch receptors. These are signals through the vagus nerve, through the nucleus tractus solitarius. And all of these are responding to satiety centers. So you have it going from this region up through to keep us from eating, and then all of this to tell us whether or not we should eat or not eat. And so you can see all of these organs are involved in satiety and stimulating us to eat as well. So the other group of neurons that get involved are the suprachiasmatic nucleus. And so you remember the suprachiasmatic nucleus is important in the timing and diurnal rhythms. And so if you wonder why rats and mice feed at night, it's because of the suprachiasmatic nucleus. So when the sun goes down and melatonin is up, that is a signal that it's time to eat. And of course, that's safety for those poor little animals in the wild, basically because they'll be less likely to be picked up by a predator. And of course, if the suprachiasmatic nucleus is destroyed and cannot respond to light or dark, then the animals will continue to feed throughout. And also interesting for us, those of us who are not nocturnal, it's nice to know that the highest leptin pulses are at night. So the leptin pulses are maximal after midnight. And that's supposed to keep us from getting up in the middle of the night and wanting a midnight snack. It may or may not work. If you're leptin resistant, it will not work. But if you're not leptin resistant, then likely you will not be wanting to eat at night. So here are some stories you can tell yourself based on what I just said. How was leptin discovered? You can talk about the parabiosis experiments, which proved that there was a factor, and making one animal skinny and keeping that animal from eating. Define orexigenic. This is where you're stimulated to want to eat. This is where you're inhibited. Where are the leptin target neurons? I would just focus in on the ones in the hypothalamus that I talked about. And then you can go through the appetite regulatory pathway. Mainly, I'm concerned, again, this is a similar question, and only the difference is, you know, what are the neurons and what do they produce? And then what are the types of signals that brain gets from the GI system? And so what are the anorexigenic, what are the orexigenic? Likely, when I give you questions I give you choices. And so you could focus in on maybe your favorite system. Maybe you really like the whole story about the GI system, for example. And so if I say, okay, tell me about how the GI system signals to the brain, then, and you can pair that to, or another question might be, tell me the route by which the adipocyte signals the brain. You could pick which system you like and focus in on the different hormones that are involved to signal the brain. And how does insulin? There's the pancreas, which is part of the GI system. And of course, it signals as we approach a meal and after we eat a meal. And then all the other things in the GI system that could stop it, for example, stretch receptors. How does this suprachiasmatic nucleus get involved? And then what about what keeps us from eating at night? And so we're non-nocturnal, but we definitely have a rhythm of leptin secretion, which will keep us from eating at night. So I think that's it. Let me get, I'm gone black. I'll stop sharing. Are there any questions? So what I would do is go through and I don't know when Dr. Saeed will ask me to submit exam questions or when you're going to get them. You may have it on your schedule, but I am happy to answer any questions about the level of detail in a question. I would rather engage it with you. Like if you don't understand a particular concept or something, and you say, I really don't understand this. Is this right? If I were to answer this question this way, would it be right? That's perfect. That's fair game. That is fair game. I'm happy to help out in that way. And so, okay. Any other questions or any questions? All right. Well, we will give you a few more minutes so you can go have lunch. So have a wonderful week, everybody.